Hello, I'm Emile Pinarbasi, a clinical instructor in neuropathology at the University of Michigan. Today, I'm going to be giving the second part of this lecture series on the neuropathologic findings associated with dementia. So first, I'm going to talk about cerebral amyloid angiopathy. This is a vascular pathology that is closely associated with Alzheimer's disease. Then I'm going to talk about three neuropathologic entities that actually do not have well-defined clinical syndromes associated with them, unlike, for example, Alzheimer's disease. So these are PART, RTAG, and LATE. All right, so we'll start with cerebral amyloid angiopathy. So if you'll recall, Alzheimer's disease is characterized by these two different pathologies. One are the amyloid plaques, and then the other is the tau tangles. So in cerebral amyloid angiopathy, this usually occurs in addition to Alzheimer's disease, although it can also occur in isolation. And instead of the amyloid accumulating within the brain tissue, it actually accumulates within the walls of the vessels. And it has a predilection for these large leptomeningeal vessels. So those are these large vessels that are in this space in between the skull and the brain, and superficial cortical vessels. So those are those are small vessels that are on the very surface of the brain, which is pictured here as kind of like this thin, dark pink ribbon that just kind of follows along the surface of the brain. So this is often just an incidental finding at autopsy. Often it's not associated with any clinical symptoms. But when there are clinical effects from this, it's usually hemorrhage or basically blood outside of the blood vessel. And this occurs because when the amyloid deposits in the walls of these vessels, it makes them kind of fragile. And so then when the vessel is exposed to any kind of stress, the vessel tears and then you get blood spilling outside the vessel. And the location of these hemorrhages kind of follows the distribution of amyloid deposition. So it's primarily kind of the very superficial surface of the brain. And this is in contrast to the other major cause of brain hemorrhage, which are hypertension-related changes. These hypertension-related changes in vessels tend to occur more in deep gray structures, the brainstem and the cerebellum, and so that's where hemorrhages caused by hypertension tend to be located. All right, so what does cerebral amyloid angiopathy look like under the microscope? So on the left is a low power view of a large leptomeningeal vessel. This is an artery, and this is an H&E, so that is our kind of standard stains that we use in pathology just to show some of the cellular detail. And in this stain, the cytoplasm of cells is usually pink and then the nucleus is blue. And now if you look over on the right, you can see there's a close-up of the vessel wall, and you can see this smooth muscle layer. And then right outside the smooth muscle layer, there's this accumulation of very bright pink material. And you'll notice it doesn't really have any structure to it. It almost kind of looks fluffy. All right, so that is a nice example of cerebral amyloid angiopathy. The pink stuff is the amyloid. And here's another image of some leptomeningeal vessels. These are a little bit smaller. And again, you can kind of see this accumulation of pink stuff in the vessel wall. And now we can also do, in addition to H&E, we can do immunostains. And this is an immunostain for beta amyloid, so it will turn all the beta amyloid brown. And what you can see is, so kind of down below is the surface of the brain, and you can see lots of these little brown patches. So these are amyloid plaques. But if you look up to the surface, you can see these brown circles, and these are these leptomeningeal vessels that have amyloid accumulating in the wall. All right, so how do we kind of write this into the pathology report? So the diagnostic line here is cerebral amyloid angiopathy. Most people will kind of comment on the severity. Most people don't use like a formal grading criteria, but they'll say something like mild, moderate, or severe. And then often this will all be grouped under all vascular changes. So if there's something like atherosclerosis or hypertension-related vascular changes, it might be in this section of the pathology report as well. All right, so moving on to primary age-related tauopathy, or PART. So PART is a very interesting neuropathologic entity. It was first described back in the 90s, but back then it was called tangle predominant senile dementia. And now it has this new name, PART, which was first coined in 2014. And this is pretty interesting. It's basically Alzheimer's disease without the beta amyloid. So if you'll remember, Alzheimer's disease is characterized by these amyloid plaques and neurofibrillary tangles. In PART, you only have the neurofibrillary tangles. But there are many different morphologies that tau pathology can take. But in PART, the tau pathology looks pretty much identical to the Alzheimer's disease pathology. And I say that in terms of distribution. So in both Alzheimer's disease and in PART, tau pathology kind of begins in the transentorhinal cortex and then progresses to involve the pyramidal neurons of the hippocampus and then finally the isocortex. And then also the molecular features of tau pathology are identical in these two diseases. So in both of these, the tau in these neurofibrillary tangles is composed of two different isoforms, 3R and 4R, which are named for the number of microtubule binding repeats that are present in the protein. And furthermore, if you do kind of advanced imaging techniques like electron microscopy, these fibrils actually have this same structure. They're these paired helical filaments. So importantly, it's very difficult to kind of parse out the clinical ramifications of PART because PART almost exclusively exists in the context of other neuropathologic diseases, like for example, FTLD or Parkinson's. However, large-scale studies do suggest that PART is associated with memory loss, which kind of makes sense given the very important role that the hippocampus plays in learning and memory. All right, so here again is an H&E of the hippocampus. So in the center is this kind of blue C, that's the dentate gyrus, and then the pyramidal neurons will kind of track along this C and then kind of spool out into the isocortex. So at low power, this looks fairly unremarkable. But when we go a little bit closer and take a closer look at some of these pyramidal neurons, you can see that they're filled with these magenta fibrils, which are actually tau. So this is the characteristic neurofibrillary tangle. And now if we do an immunostain for tau, you can see that all of these neurons are lighting up. And if we use immunostains that are specific for these two isoforms, the 3R and the 4R, we can see that in fact both of them are positive, which tells us that these neurofibrillary tangles have both isoforms of protein inside them. So PART is staged very similar to the way, or exactly the same as the tau pathology within Alzheimer's disease. We use this staging system called Brock staging. So people have described that tau pathology begins in the entorhinal cortex and then progresses in this stereotyped manner throughout the hippocampus and then finally involves the isocortex. And so in Brock staging, you basically look at the hippocampus, you do an immunostain for tau, and then you kind of picture match the degree of tau pathology to the extent of tau pathology in these different stages. So in this case over on the left, you can see that there is quite a bit of tau pathology in the entorhinal cortex and also in CA2 and CA1. So this is pretty good for stage 3. And importantly, you actually don't see part progressing past Brock stage 4. The reasons for this are not entirely clear, but the most recent line of thought is that in order for the tau pathology to kind of escape past the hippocampus and into the isocortex, beta amyloid is required. All right, and so how does this appear in a pathology report? So basically you have your diagnosis, primary age-related tauopathy or PART, and then because this diagnosis is actually can be very challenging to differentiate from other tauopathies, and also because this almost always occurs in the context of other neuropathologic diseases, we also remark whether it's definite or indefinite PART. And then finally we give a Brock stage. All right, so moving on to another tauopathy, age-related tau astrogliopathy, or RTAG. So RTAG, unlike PART, is an accumulation of tau within astrocytes as opposed to neurons. Also, instead of being composed of two isoforms, 3R and 4R, it's simply the 4R isoform. And it's characterized by this very specific morphology, which I'll show you, and a specific distribution. It's usually seen in these very striking little clusters, and it almost always involves the medial temporal lobe, in particular the amygdala. The clinical significance of RTAG is really not clear. It's probably kind of the least likely to have any clinical significance of any of the entities that I talk about in this video. So what I'm showing here is an image that compares some of the morphology of tau in astrocytes in the primary tauopathies, which include things like CBD, PSP, MSA, or sorry, CBD or PSP versus what we see in RTAG on the bottom. And what you can see is that in RTAG, instead of having these kind of like long processes of tau coming out from astrocytes, you have what we call thorny astrocytes, where the tau is just like mostly in the cell body. And then there's this other morphology where they're kind of like fuzzy, instead of like these well-formed projections. All right, so this is a very classic look for RTAG. What you're looking at here is the inferior surface of the brain. You can see some vessels at the bottom, and then you can see the peel surface of the brain. And then right above that, you see these kind of clusters of tau staining. And here is another image of RTAG. Here we have in the middle of the screen a vessel, and then now we have these clusters of thorny astrocytes that are kind of right around this vessel. So this is the paravascular pattern. And then finally, now we're looking at the amygdala, again, the tau immunostain. And there's this white matter tract that delineates the amygdala, and you can see that there's this sort of line of brown, this line of tau stain that perfectly follows that white matter tract. And again, it's these thorny astrocytes. And so this is a good image, a good example of the white matter pattern of RTAG. And here's just a close-up showing the morphology in these cells, these little thorny astrocytes. All right, so how does this appear on a diagnostic line? So it begins with the diagnosis, age-related tau astrogliopathy or RTAG. Then we always comment on the type of RTAG or the pattern. So in this case, I've showed you subpeel, paravascular, and white matter patterns. We always comment on the location. So in this case, it was all in the medial temporal lobe. And then we also comment on whether it's focal or widespread. All right, and then finally, the last neuropathologic entity I'll discuss is LATE, or limbic predominant age-related TDP-43 encephalopathy. So this is a pathology that is actually quite common. If you look in the population of individuals greater than 80 years old, which we've termed, quote unquote, the oldest old. So in this pathology, you see an accumulation of this protein, TDP43, in both neurons and in glia. And particularly, you see this accumulation in the medial temporal structures, the amygdala, the hippocampus, and then it eventually spreads out into the frontal cortex. One interesting thing about late is that it's pretty much exclusively seen in the context of tauopathy. This is usually Alzheimer's disease, but it can be seen in things like CBD, PSP, and PARP, which I talked about earlier. It's closely associated with another pathology that I'll describe briefly called hippocampal sclerosis. And like the other neuropathologic entities I've discussed today, the clinical significance is very difficult to determine because this, exists in the context of other neuropathologic diseases, but large-scale studies suggest that it is associated with an amnestic dementia. All right, so what does this one look like under the microscope? Again, here we have an image of the hippocampus. Here we have the dentate gyrus, which is like that blue C. And then again, the pyramidal neurons kind of spooling out. This is an immunostain for phospho-TDP43, which will highlight the aggregates. And you can see here at low power that there's quite a bit of brown staining in CA2, CA1. And then when you take a close-up look, you can see some of these little kind of round brown dots in neurons. So those are the aggregates. You also see this pathology in the amygdala. So here's an image of the amygdala at low power. It's really difficult to appreciate that there's any staining, but when you take a closer look, you can see some neurons with these really clear aggregates. And then finally, when this pathology progresses, it can involve the frontal cortex. So this is, again, the immunostain for phospho-TDP43 in the frontal cortex. And at low power, very difficult to see any staining, but when you go at higher power, you can appreciate that there are a lot of aggregates in both neurons and glia. So as I've kind of alluded to, late pathology is known to sort of begin in the amygdala, progress to involve the hippocampus, and then finally, in the latest stages, involve the frontal cortex. And so we stage this disease by taking standard sections of all these brain regions and then doing an immunostain for phospho-TDP43 and then assessing for the presence of aggregates. And then one final note, this pathology, as I mentioned, is often associated with something called hippocampal sclerosis. So hippocampal sclerosis is basically the pathologic description that we have for a hippocampus that is kind of so diseased that you have, that it basically looks scarred and has kind of like a white, firm, rubbery appearance if you just like look at the brain grossly, and then under the microscope you can appreciate that you have dropout of neurons. So here again you have an H&E of the hippocampus, and you'll notice that the dentate gyrus, instead of being sort of this continuous blue line, you can see it almost looks like someone took an eraser and just kind of smudged out areas. And so that's actually sections where the neurons have dropped out. And you can highlight this by doing an immunostain for nu-N, which will stain all the neurons. And so again, instead of seeing a sort of continuous line of neurons, you can see neuronal dropout, basically due to death, in this case, due to this accumulation of tau pathology. That's basically killed whole sections of neurons. All right, so how will you see this in a pathology report? So again, we see the diagnosis. limbic predominant age-related TDP-43 encephalopathy, or late NC. We always give the stage. In this case, I showed you something that was stage 3. And then finally, people comment on the presence or absence of hippocampal sclerosis. All right, so just to briefly review, today I talked about cerebral amyloid angiopathy, a vascular pathology that's associated with Alzheimer's disease, and then I discussed 3 neuropathologic entities: PART, RTAG, and late. And thank you for your attention.